Mitotic CDK complexes are maturation promoting factor MPF or M phase promoting factors are synthesized during the S phase and G2. Mitotic CDK complexes induce chromosome condensation, breakdown of the molecular envelope, assembly of the mitotic spindle apparatus, and alignment of condensed chromosomes at the metaphase plate. After the proper association of all chromosomes with the spindle microtubules has occurred, the mitotic CDK complex activity anaphase promoting complex or APC. This multi-protein complex is a ubiquitin ligase that directs the ubiquitin-mediated proteolysis of anaphase inhibitors, leading to inactivation of the This multi-protein complex is a ubiquitin ligase that directs the ubiquitin-mediated proteolysis of anaphase inhibitors, leading to inactivation of the protein complexes that connect sister chromatids at metaphase. Then degradation of these inhibitors, this permits the onset of anaphase. The destruction of cyclin is as important for exist from mitosis as its synthesis is for entry. The APC also directs proteolytic degradation of the mitotic cyclins. Then mitotic cyclin ubiquitinization is performed by APC, which is activated in late mitosis by the addition of a CDC20. APC contains binding sites that recognize specific amino acid sequences of the target protein. Then CDC20 is there, APC inactive. Mitotic cyclin is forming CDK, then APC is formed that turns active. Then ubiquitin mediated proteasomal degradation of mitotic cyclin happens. Then the resulting decrease in mitotic CDK activity permits the separated chromosomes to decondense and nuclear envelope to reform around daughter cell nuclei during telophase and the cytoplasm to divide at cytokinesis, yielding the two daughter cells. Then passage through three critical cell cycle transitions, G1 to S phase, metaphase to anaphase, and anaphase to telophase and cytokinesis, is irreversible because these transitions are triggered by the regulated degradation of proteins. As a consequence, cells are forced to traverse the cell cycle in one direction only. Then regulation of activity of cyclin CDK complex. The rise and fall of cyclin levels in the primary determinant of CDK activity during the cell cycle. The protein kinase activity of the cyclin CDK complex also depends on the phosphorylation state of the catalytic CDK subunit. The CDK activity is inhibited by protein tyrosine kinase V1 catalyzed phosphorylation of tyrosine. This inhibitory phosphate is removed by the CDC25 protein phosphatase. A decrease in WEE activity and increase of CDC25 activity resulting in activation of cyclin CDK complex. Full activation of the cyclin CDK complex occurs when a separate kinase, the CDK activating kinase CAK, phosphorylates an activating threonine residue near the CDK active site. This causes a small conformational change that further increases the activity of the CDK, allowing the kinase to phosphorylate its target proteins effectively. Then regulation of activity of MCDK complex in S-FOP. The interaction of mitotic cyclin with cyclin dependent kinase forms MCDK complex. The CDK subunit can be phosphorylated at two regulatory sites by V1 at tyrosine 15 and CAK in threonine 161. Removal of the phosphate on tyrosine 15 by CDC25 phosphatase yields active MCDK complex. Then mitotic cyclin is there, CDK is there, MCDK inactive, V1 CAK, then MCDK inactive form. Inhibitory phosphatase is there, activating phosphatase is there, CDC25 P15 MCDK active form and P161. Then cyclin CDK complexes can also be regulated by the binding of CDK inhibitor proteins, CDKIs or CKIs. CKIs binding make the cyclin CDK complex inactive. There are variety of CKI proteins like P25, P21, P16, and they are primarily employed in the control of G1 and S phase. Then cell cycle checkpoints. The cell cycle is a highly regulated and a dependent series of events mediated by a number of checkpoints. Cell cycle checkpoints function to ensure that incomplete or damaged chromosomes are not replicated and passed on to daughter cells. In most cells, there are several checkpoints in the cell cycle at which the cycle can be arrested if previous events have not been completed. There are three major checkpoints in the cell cycle: G1 checkpoint towards the end of G1 phase, then G2 checkpoint towards the end of G2 phase, then metaphase checkpoint, M phase. In most of the cell, the major cell cycle checkpoints is in late G1, commitment to chromosome replication. This G1 checkpoint is called start in yeast cells or a restriction point in animal cells. G1 checkpoint blocks progression into S phase. G2, called the mitotic entry checkpoint, is most important in fission yeast. Then role of RP protein in cell cycle regulation. RP protein is a product of retinoblastoma gene, a tumor suppressor gene. Genes whose inactivation contributes to the development of a tumor are called tumor suppressor genes. Loss of function mutations of RB genes are associated with the disease hereditary retinoblastoma. The RB gene is located on chromosome 13q14.2. The RB protein encoded by the retinoblastoma gene is a 105 KDA polypeptide gene, also known as P10FRB, which can be phosphorylated. How the product of RB gene regulates cell division. RB is a substrate for CDK cyclin D complexes and exerts its effects during the part of G1 that precedes the restriction point. It acts as a brake in mammalian G1 cells. E2F, a transcription factor, binds to specific DNA sequences and induces many types to transcribe. High levels of E2F activate transcription of cyclin A gene. The products of these genes are required for S phase of cycling. Then E2F activity is controlled by RB protein. When RB protein binds to E2F, its activity is suppressed and transcription of gene involved in S phase stops. Phosphorylation of RB protein, reducing its affinity for E2F and allow E2F to activate S phase gene expression. Phosphorylation of RB protein is initiated by cyclin D-CDK4 and cyclin D-CDK6 in mid G1, and or hypophosphorylated RB proteins block cell cycle progression. In homozygous null RB genes cells, RB protein is permanently inactive. This E2F is always able to promote S phase, and the arrest of normal cells in late G1 does not occur in retinoblastoma cells. Role of P53 in cell cycle regulation. DNA damage blocks progression through the cell cycle until the damage is repaired. Damage to DNA can result from chemical and physical agents. Tumor suppressor protein P53 contribute to arrest of cells with damaged DNA at DNA damage checkpoint. Cells with functional P53 arrest in G1 and G2, whereas cells lacking functional P53 do not arrest in G1. And P53 gene codes for P53 protein as a well-characterized tumor suppressor gene, so named because it encodes a phosphorylated protein with an apparent molecular weight of 53 kilodalton. It is located on human chromosome 17p13.1 and encodes a polypeptide chain of 375 amino acid residues. P53 protein is a transcription factor that regulates the expression of cell cycle regulatory proteins. P53 is not required for normal cell division. Thus, under normal conditions, it is extremely unstable. The proteasomal degradation of P53 results from its polyubiquitination by a ubiquitin ligase called MDM2. In case of DNA damage, the rapid degradation of P53 is inhibited by ATM, which phosphorylates P53 at a site that interacts or interferes with binding by MDM2. In response to DNA damage, P53 levels increase and arrest the cell at the G1 phase of the cell cycle. If all of the repairs have been made to the DNA, the cell divides normally and completes the cell cycle. However, if the cells still contains mutated or duplicated DNA sequences, it dies by a suicidal apoptotic mechanism to prevent its expansion. In those cells that have mutated or lost p53 gene, the arrest of G1 does not occur, and the cells that have mutated genomes proliferate and become cancerous. So MDM2 p53 is there, damaged DNA, activation of ATM like serine kinase, then phosphorylation of p53, MDM2 p53 just releases there, and that becomes activated, act as transcription factor for p21 gene. Then synthesis of p21 protein acts on CKI, binds with SCDK, then SCDK is active, then SCDK inactive. How DNA damage arrests the cell cycle in G1. That's what represented by this figure. Next topic, cell division. In eukaryotes, two types of cell division is partition the genetic material into offspring or daughter cells in one process called mitosis. Parent cell divides into two daughter cells and each receive an exact copy of the genetic material on the parent cell. In the other partitioning process, the genetic material is must precisely half so that fertilization will restore the diploid complement. The cellular process is called meiosis. What is mitosis? Mitosis is a process that partitions newly replicated chromosomes equally into two daughter cells. The term mitosis derived from the Greek word meaning thread. Thread was introduced by Walter Fleming in 1882. During mitosis, one round RNA replication is followed by a single round of chromosome segregation and generating two generally identical daughter cells. Mitosis is a continuous process. They are conventionally divided into four stages like prophase, metaphase, anaphase, and telophase. Then prophase. The chromatin, which diffuses in interphase, slowly condenses into well-defined chromosomes. Each chromosome has duplicated during the preceding S phase and consists of two sister chromatids. Each of these contains a specific DNA sequence known as centromere, which is required for proper segregation. At this point, sister chromatids are kept together by a complex called cohesin, made up of at least four different proteins. Prophase ends abruptly with disruption of nuclear envelope, which breaks into membrane vesicles that are indistinguishable from bits of endoplasmic reticulum. These vesicles remain visible around the spindle during mitosis. At this time, also the nucleolus disappears. Toward the end of prophase, the cytoplasmic microtubules that are part of interphase cytoskeleton disassemble and the main component of the mitotic apparatus, the mitotic spindle, begins to form. This is a bipolar structure composed of microtubules and associated proteins. The spindle initially assembles outside the nucleus within separating centrosomes. So replicated chromosome is there, centromere region of chromosome is there, kinetochore is there, then kinetochore microtubules are associated with that, then chromatid region is also there. So the mitotic spindle at metaphase is of three types: kinetochore microtubule, which attach with chromosomes at specialized attachment sites on the chromosomes called kinetochores, and the kinetochore is a specialized attachment site at the chromosome centromere. The sister chromatids of metaphase chromosome are transported to each pole along the kinetochore microtubule. Astral microtubules, which forms the aster, they radiate outward from the centrosome toward the cortex of the cell. Polar microtubules, kinetochore microtubule, astral microtubule, and polar microtubules do not interact with chromosomes but instead interdigitate with polar microtubules from the opposite pole. Then kinetochore microtubule is there, astral microtubule is there, centrosome is there, polar microtubules are there. So in case of metaphase, in the beginning of metaphase, specialized protein complexes called kinetochores mature on each centromere and attach to some of the spindle microtubules, which are then called kinetochore microtubules. The remaining microtubules in the spindle are called polar microtubules, while those outside the spindle are called astral microtubules. The kinetochore microtubules align the chromosomes in one plane halfway between the spindle poles. Each centromere is held in tension at this metaphase plate by the paired kinetochores and their associated microtubules, which are attached to opposite poles of the spindle. Then assembly of the metaphase spindle requires two types of events: attachment of spindle microtubules to the poles and capture of chromosomes by kinetochore microtubules. A kinetochore contacts the sides of the pole and capture the chromosome by kinetochore microtubule. A kinetochore contacts the side of a microtubule and then slide along the microtubule to the plus end in a process that may involve kinesins in the kinetochore. The kinetochore caps the plus end of the microtubule. A combination of microtubule motor proteins at the kinetochore and microtubule dynamics of the plus end of kinetochore microtubules is thought to position the chromosomes equally between the two spindle poles. Here we could see the picture. The next stage, anaphase. The next phase, anaphase, as mentioned, each sister chromatid of a metaphase chromosome is attached to microtubules via its kinetochore, a complex of proteins assembled at the centromere. The opposite ends of these kinetochore microtubules associate one of these spindle poles. At anaphase, the kinetochore spindle tubers pull the two kinetochores toward the opposite poles. Then sister chromatids do not separate because they are held together at their centromeres by multi-protein complexes called cohesins. Cohesins associate with chromatin at S phase. During anaphase, separase, a ubiquitinous protease, degrades cohesins. A securin protein keeps separates in an inactive state. The destruction of securin releases separase, which is then free to cleave cohesin complex. Destruction of securin is mediated by anaphase promoting complex APC, ubiquitin ligase. Each chromatid, now called a chromosome, to be pulled slowly towards the pole at phases. Then securin is there, separate or inactive form. Later degraded securin is there, separase is there, and in the active form. So the separation of sister chromatid as a result of proteolysis of cohesin. The destruction of securin allows separase to cleave a subunit of cohesin. Then all of the newly separated chromosomes move at the same speed, typically about 1 mm per minute. The chromosomes move by two independent and overlapping processes. Hence, anaphase can be divided into two distinct stages like anaphase A and anaphase B, or early and late anaphases. Anaphase A is characterized by the shortening of kinetochore microtubules, which pulls the chromosomes toward the poles. Microtubule shortening during anaphase A is due to depolymerization of kinetochore, preferentially to the plus ends of microtubules. During anaphase B, the two poles move further apart, and it happens as a result of spindle elongation, which occurs due to sliding of polar microtubules past one another and pulling forces exerted by astral microtubules. Like anaphase A is there shortening of kinetochore microtubules, movement of daughter chromosomes to poles, forces generated mainly at kinetochore. Then anaphase B, a sliding force is generated between overlap microtubules from opposite poles to push the poles apart. A pulling force acts directly on the poles to move them apart. The microtubule growth at plus and end of polar microtubules. The separation of daughter chromosomes at anaphase A and B in animal cells. Adapted from Alberts et al., the Cell, 4th edition. So then that's the picture. Then telophase, next stage is telophase. In telophase, the separated daughter chromosomes arrive at the poles and the kinetochore microtubules disappear. The polar microbodies elongate still more and a new nuclear envelope reforms around each group of daughter chromosomes. The condensed chromatin expands once more, and nucleoli which had disappeared at prophase begin to reappear, and mitosis is at an end. Then cytokinesis. The cell cycle culminates in the division of cytoplasm by cytokinesis in a typical cell. Cytokinesis accompanied every mitosis, although some cells such as Drosophila embryos undergo mitosis without cytokinesis and become multinucleate. Cytokinesis begins in anaphase and ends in telophase, reaching completion as the next interphase begins. Then cytokinesis in animal cells. Cytokinesis appears to be guided by organized bundles of actin filaments in animal cells. During cytokines and cytoplasm divides into a process called cleavage. Cleavage is accompanied by contraction of circumferentially oriented filaments, actin filament and myosin 2, bound to the cytoplasmic phase of the plasma membrane. This contractile ring defines the cleavage of the muscle-like sliding of actin and myosin 2 filaments, and the contractile ring generates the force required for cleavage. This is the picture of contractile ring. Then next stage, cytokinesis in plant cells. Cytokinesis occurs by a special mechanism of higher plant cells where the cytoplasm is partitioned by the construction of a new cell wall, the cell plate inside the cell. A cytoskeleton framework determines the plate of plant cell division. A array of microtubules appear in G2 phase and forms a ring around the entire cell just beneath the plasma membrane. This is called prophase band. It also contains numerous actin filaments in addition to microtubule. The microtubule disappears before metaphase, but the actin filaments remain and provide a memory of the predetermined division plane. The polar spindle microtubules assemble in this plane and forms a cylinder-like structure called phragmoplast. Small membrane-bound vesicles derived from the Golgi apparatus and filled with cell-walled precursors associate with the microtubule and form the early cell plate. The polysaccharide molecule derived by these vesicles forms the matrix polysaccharide. Sometime later, cellulose microfibrils are laid down within the cell plate to complete the new cell wall. The next topic, meiosis. Meiosis coined by Farmer and Moore. This is a specialized form of cell division in which number of chromosomes are reduced by half. The reduction in chromosome number is achieved by one round of DNA replication being followed by 2 rounds of chromosome segregation with no intervening round of DNA replication. Meiosis is divided into 2 parts like meiosis 1 and meiosis 2. At the end of meiotic process, there are 4 daughter cells rather than 1. The 2 produced at the end of meiotic process, each of which resulting daughter cells has 1/2 of the number of chromosomes as the parent cell. Then meiosis 1. Prophase 1 of meiosis 1 is divided into 5 substages: leptotene, zygotene, pachytene, diplotin and diakinesis. So first, leptotene phase. Prophase 1 begins at the leptotene stage when each chromosome is first seen to have condensed from its interphase conformation to produce a long thin thread with a proteinaceous central axis. Each chromosome is attached at both of the ends nuclear envelope by a specialized structure called the attachment plaque. Although each chromosome has replicated and consists of 2 sister chromatids, these chromatids are unusually closely opposed, and each chromosome therefore appears to be single. Separate chromatids will not become visible until late in prophase at either the diplotin stage or in diakinesis. In the leptotene to zygotene transition, the tips of the chromosome move until most end up in a limited region near each other. The forms arrangement called a bouquet stage. Then next stage, zygotene. Leptotene is considered to end and the zygotene stage of prophase to begin as soon as synapsis or intimate pairing between the 2 homologous portions is initiated. The initial recognition requires that the homologous recognize each other from a distance. How the maternal and paternal copy of the chromosomes recognize each other and is still unknown. Synapsis often starts when the homologous ends of the 2 chromosomes are brought together on the nuclear envelope and continues inward in a zipper-like manner from both ends. Synapsis often starts when the homologous ends of the 2 chromosomes are brought together on the nuclear envelope and continues inward in a zipper-like manner from both ends, aligning the 2 homologous chromosomes side by side. In other cases, synapsis may begin in internal regions of the chromosome and proceed toward the ends, producing the same type of alignment. Each gene is thus brought to be thought into juxtaposition with its homologous gene on the opposite chromosome. As the homologous pair, their rock-like proteinaceous axes are brought together to form the 2 lateral elements or sides of the long ladder-like structure called the synaptonymal complex. Each resulting chromosome pair with meiotic prophase 1 is usually called a bivalent, but since each homologous chromosome in the pair is a tetrad, another commonly used term. Here we could see central element of synaptonemal complex is there, then paternal sister chromatids, chromatid 1 and chromatid 2, then maternal sister chromatids, chromatid 1 and chromatid 2, and protein axis lateral elements are there. Then next stage packaging. As soon as synapsis is complete all along the chromosomes, the cells are said to have entered the packaging stage or prophase where they may remain for days. At this stage, large recombination nodules, large protein complexes, contain RAD51 karyotic version of Rec A protein appear at intervals on the synaptonemal complex and are thought to mediate crossing over. These exchanges result in crossovers between two non-sister chromatids, that is, one from each of the two paired homologous chromosomes. Although invisible at packaging, each such crossover will appear later as a chiasma. Then it is a physical exchange between non-sister chromatids in a pair of homologous chromosomes. The exchange process consists of breaking and rejoining of two chromatids. It is the reciprocal exchange of equal and corresponding sequel from them. Direct cytological evidence that homologous chromosomes exchange part during crossing over has obtained in 1931 by Stern working with Drosophila and by H.B. Crichton and B.M.C. McClintock working on maize. Crossing over occurs more or less random along the length of a chromosome pair. The crossing over occurs in the tetrad stage, bivalent or packaging stage of meiosis 1. The crossing over may occur also in somatic cells as reported by Stern in Drosophila. Male Drosophila and female silkworm Bombyx mori are examples of organisms in which crossing over does not occur in meiotic cell. So that's a peculiarity. Male Drosophila and female silkworm Bombyx mori are examples of organisms in which crossing over doesn't occur in meiotic cell. Then diplotin. Desynapsis begins the diplotin stage of meiotic prophase 1. The synaptonemal complex dissolves, allowing the two homologous chromosomes in a bivalent to pull away from each other to some extent. However, each bivalent remains joined by one or more chiasmata representing the sites where crossing over has occurred. In new sites or developing eggs, diplotin can last for months or years, since it is at least this stage that the chromosome decondenses and engage in RNA synthesis to provide storage materials for the egg. In the extreme, the diplotin chromosomes can become highly active in RNA synthesis and expand to the enormous extent, producing the lamprey chromosomes found in amphibians and in some other organisms. Then diakinesis, last process. Diplotin merges imperceptibly into diakinesis, a stage of transition to metaphase, as RNA synthesis ceases and the chromosome contents begin and become detached from the nuclear envelope. Each bivalent contains 4 separate chromatids, with each pair of sister chromatids linked at their centromeres. After prophase, cell enters into the metaphase 1 and followed by anaphase 1. During anaphase 1, segregation of homologous chromosomes occur, like meiotic prophase 1, then meiotic anaphase 1, then meiotic metaphase 2 stage, then meiotic metaphase 2 stage after that. So metaphase 1, anaphase 1 and telophase 1 is there. So metaphase 1 is marked by the breakdown of nuclear membrane and the attachment of kinetochore microtubules to the bivalent. During metaphase 1, the homologous chromosomes move to the equatorial plate called the metaphase plate. Unlike in meiosis, in which sister chromatids are pulled apart, in anaphase 1, homologous chromosomes are separated. Homologous chromosomes separate and move to opposite poles. This meiotic division is therefore called a reductional division because it reduces the number of chromosomes to half the diploid number of each daughter cell. Crossing over and independent assortment are responsible for genetic variability during meiosis. Crossing over between homologous chromosomes and cause assortment of genes in individual chromosomes. Independent assortment of maternal and paternal homologies occur during the metaphase at first meiotic division. It produces second two and different haploid gametes for an organism with n chromosomes. Then meiosis 2 is there. Meiosis 2 is similar to mitosis. However, there is no S phase. The chromatids of each chromosome are no longer identical because of recombination during meiosis 1. Meiosis 2 separates the chromatids, producing two daughter cells, each with haploid chromosome number. Then non-distinction and aneuploidy. During mitosis, each chromosome duplicates and the two daughter chromosomes separate, with one going to each progeny cell. Similarly, during meiosis, the two homologous chromosomes separate during the first division, and then the two daughter chromosomes, previously chromatids, move to separate cells during the second division. The result of a normal mitosis or meiosis is that all the progeny cells receive a complete set of chromosomes. Occasionally, however, homologous chromosomes and daughter chromosomes fail to separate or disjoin properly during meiosis or mitosis. The failure of chromosomes to separate properly during mitosis or meiosis is called non-distinction. Non-distinction produces cells or gametes with extra or missing chromosomes. It is the primary case of aneuploidy. Then segregation and independent assortment during meiotic division. In an organism that is heterozygous for two different alleles of a gene, each such as tall and tough, in Mendel's piece, the two alleles segregate from each other during the formation of gametes. The biological basis of the segregation is the pairing and subsequent separation of homologous chromosomes during the first. Segregation is the pairing and subsequent separation of homologous chromosomes during the first or a reductional division of meiosis. Then separation of homologous chromosomes during the first division of meiosis. Pairing of homologous chromosomes in the first division of meiosis is there of orientation. Gametes are produced. New combinations of genes. New combinations of genes on non-homologous chromosomes are produced by the independent assortment of chromosomes during the first division of meiosis. Consider the cross between two double homozygotes, for example AABB and AABB, where genes A and B, capital B, are on different chromosomes. The F1 progeny, F1 for first filial generation, will be double heterozygotes. Half of gametes produced by F1 progeny will have the same combination of alleles as the parents. The other half will have new recombinant combinations of alleles. The combinations of pairs of independent assortment of homologous chromosomes during the first division of meiosis. Each pair of homologous chromosomes lines up at the metaphase plate independently of every other pair. Both maternal chromosomes may be on the same side of the metaphase plate with the two parental chromosomes on the other side, or one maternal chromosome may be on the same side of the metaphase plate as the parental member of the other chromosome pair, and vice versa. The result is that the segregation of alleles of a gene on one chromosome occurs independently of the segregation of alleles of any other on a non-homologous chromosome. Comparison between mitosis and meiosis in animals. Mitosis occurs in all tissues, but meiosis occurs only in testis and ovary. Produces diploid cells in case of mitosis, but meiosis produces haploid cells or gametes. Then DNA replication, one round of replication per cell division, but only one round of replication, but two cell divisions in case of meiosis. Then extent of prophase is short in case of mitosis, but it's long in meiosis. Then pairing of homologies is absent in mitosis but present in meiosis. Then recombination is rare in mitosis but common in meiosis. Relationship between daughter cells just genetically identical but it's different in case of meiosis. Then next topic, apoptosis. Apoptosis is from the Greek words apo, from, and ptosis means falling. It's a programmed cell death. It is a process of deliberate suicide by the unwanted cell in a multicellular organism during embryogenesis, morphogenesis, or metamorphosis and tissue turnover. It is the capacity of a cell to respond to a stimulus by initiating a pathway that leads to its death by a characteristic set of reactions. In contrast to necrosis, which is a form of cell death that results from the acute tissue injury, apoptosis is carried out in an ordered process that generally confers advantages during an organism's life cycle. For example, the differentiation of human fingers in a developing embryo requires the cells between the fingers to initiate apoptosis so that the fingers can separate. The demise of cells by programmed cell death is marked by a well-defined sequence of morphological change, apoptotic cells become more compact, blubbing together at the membranes. Chromatin become condensed, DNA is fragmented, dying cells shrink and condense and then fragment, releasing small membrane-bound apoptotic bodies, which generally are phagocytosed by surrounding cells. Importantly, the intracellular constituents are not released into the extracellular milieu where they might have deleterious effects on neighboring cells. The highly stereotyped changes accompanying apoptosis suggest that this type of cell death is under the control of a strict cellular program. Then apoptosis can be triggered by a variety of stimuli, including withdrawal of essential growth factors, treatment with glucocorticoids, gamma irradiation, and activation of certain receptors. Another means of initiating apoptosis is used in the immune system, where cytotoxic T lymphocytes attack target cells. The engines of cell destruction are a series of enzymes called caspases, so named because they contain a key cysteine residue in the catalytic site and selectively cleave proteins at sites just C-terminal to aspartate residue. The caspase 3 subfamily consisting of caspase 3 and caspase 6 to 10 is involved in apoptosis. All the caspases are synthesized in the form of inactive procaspases, and each caspase, when activated, cleaves certain target proteins at specific aspartate residues in the target polypeptide genes. There are multiple apoptotic pathways. While caspases are critical in many apoptotic events, caspase-independent apoptosis also occurs. Like caspase-independent extrinsic apoptotic pathways, their killer lymphocytes can induce apoptosis by extracellular activation. They induce apoptosis by producing a protein called Fas ligand, which binds the death receptor protein Fas on the surface of target cell. Fas is a cell surface receptor. The Fas ligand is a transmembrane protein. The Fas receptor on the target cell is activated by interaction with the Fas ligand on the activated cell plasma membrane. Fas has a cytoplasmic domain called the death domain, which is involved in protein-protein interactions. Fas proteins then recruit intracellular adapter proteins that bind and aggregate procaspase 8 molecules, which cleave and activate one another. The activated caspase 8 molecules then activate downstream procaspases to induce apoptosis. Then killer lymphocyte is there, Fas ligand, adapter protein, target cell, activated procaspase 8, then activated caspase 8, apoptosis of target cells, caspase cascade. A killer lymphocyte carrying the Fas ligand binds and activates Fas proteins on the surface of the target cell. Adapter proteins bind to the intracellular region of aggregated Fas protein, causing the aggregation of procaspase 8 molecules. These then cleave one another to initiate the caspase cascade. Then caspase independent intrinsic apoptotic pathway. The mitochondria plays a central role in apoptosis by releasing cytochrome C. The basic role of cytochrome C is to trigger the activation of caspase 9. Cytochrome C triggers the interaction of cytosolic adapter protein APAF1 in procaspase 9 in a complex called apoptosome. The incorporation of procaspase 9 into the apoptosome triggers the auto-activating cleavage. Caspase 9 in turn cleaves procaspase 3 to generate caspase 3, which cleaves targets that cause apoptosis of the cell. Then injured mitochondrial release of cytochrome C, then bind with APAF1 that form binding with procaspase 9, then activation of procaspase 9. So schematic representation of the role of mitochondria in apoptosis. Cytosolic cytochrome C interacts with APAF1, allowing subsequent caspase activation. Then apoptosis regulatory proteins. Regulatory proteins comprises both anti-apoptotic membranes and pro-apoptotic membranes. BCL2 was the first oncogene shown to mediate its effects on the inhibition of apoptosis. It is the mammalian homoplague of CED9 gene in C. elegans. Anti-apoptotic members like BCL2, BCLXL inhibit apoptosis. The exact mechanisms by which the anti-apoptotic members regulate apoptosis remain elusive. Similarly, BAX, BAD, BIC act as a positive regulator. So that's all about this topic. Next topic answer, which will be dealt with the help of another video. Thank you for listening.